everyone, Yamina here. Welcome to the Dr. GPCR Podcast. Today's episode dives into persistence, passion, and the custom paths that truly accelerate drug discovery. I'm joined by Dr. Sokhom Pin, Head of Biology at Superluminal Medicines, whose GPCR journey spans decades across BMS, Novartis, Alkermes, and Cerevel. Before we go deeper, while Sokhom and I were colleagues at Superluminal, I am no longer with the company. What makes Sokhom's story stand out isn't just his deep expertise into GPCR pharmacology. It's how he earned a PhD while working full-time and supporting a growing family. In this episode, you'll hear how Sokhom turned personal constraints into a custom academic industry PhD, how he built the happiest team at Alkermes, and why culture matters just as much as science does in drug discovery. And if you're looking to deepen your own expertise, head to Terry's Corner now, the only expert-led, on-demand pharmacology corner. We release 4 new courses every month and host regular Ask Me Anything sessions with Dr. Terry Kinakin himself. Learn more today at terrykinakin.com. You'll find all other relevant links, including our featured partner, Celtaris Research, in the description of this episode. And now, let's hear from Sokhom.
Hello, everyone. This is Yamina for Dr. GPCR. And this evening, I have a very special guest. Sokhom and I connected in 2021, I think, on LinkedIn. And up until very recently, we didn't have a chance to chat. And today, I'm very excited to have him with us as our guest on the Dr. GPCR podcast. Sokhom is currently a head of biology at Cerevel Therapeutics, which was acquired by AbbVie. Sokhom, welcome to the podcast.
Thank you, Yamina. Thank you for having me on the show.
I'm very excited to have you on. Could you please introduce yourself to the audience and tell us who you are and what is it that you do?
So I'm Sokhom Pin, and basically all my life, I'm an industry scientist. I started my career at DuPont Pharmaceuticals in high-throughput screening departments, and then 2 years later, then DuPont was bought up by BMS, and then I joined BMS as part of the neuroscience department. And that's where I started working mostly on GPCRs, classes of targets. And then 8 years at Novartis. So 8 years at BMS, 8 years at Novartis, and another 4 years at Alkermes, and 5 years at Cerevel. And now it's been 4 months at AbbVie, and then Superluminal. So all my entire industry experience all been GPCR focused.
I love it. Did you always know you wanted to be a scientist?
Well, actually, I knew when I came to the US and even in middle school that I wanted to have a doctoral degree. But it only after college that I decided the PhD route to be a scientist.
Interesting. And while in middle school or in college, was there anything specific you're interested in? The reason I'm asking is we've had guests who said, oh, biology was it because I had this great biology teacher, or some had said actually astrophysics and they ended up in biology, but I'm curious on your end.
Yeah, so basically my typical Asian family that the only profession that my parents knew was being a medical doctor. So that was my aim from the beginning. So, and then I was majoring in biology, and it's not until I started working as a technician at Johns Hopakins Hospital, where I worked with MD and PhDs, and worked with a lot of postdocs who were MD and who were PhDs. So when I made the comparison, look at their lives as the MDs versus the PhDs, I decided that I think I wanted to be a PhD rather than MD.
Interesting. I think I'm also in that bucket where the parents wanted to have another MD in the family. And I actually spent some time volunteering at Saint Justine Hospital. It's a children's hospital that has a research center. So I was doing my graduate studies there. And I think after 2 times volunteering and working with sick children, I realized that as much as I do love kids, I cannot be an MD. I have to focus on the science and potentially try and find, medicines to help kids.
Yeah, so at Johns Hopkins, I was so excited about every time I have my name on publications. So I was just so passionate about trying to get my name on a publication.
Well, speaking of publications, and it's a really funny thing. So yesterday I recorded a podcast episode with my postdoc mentor, Tom Sackmore, and Ilana Kotliar, who just finished her PhD in the lab. And before we started recording, we were on PubMed because 9 years ago, I collaborated with a lab at NIH. And the paper finally got published in September of this year. And it's a science paper. We started out with 6 authors. We're now 95. And with all the hashtags and asterisks, yesterday when we were looking at it with Tom, I realized that I'm co-first author with 5 other people. And I know that I'm not in academia anymore, but it made me feel so good knowing that I have a co-first author in authorship paper in science.
That's awesome. Great. Congratulations.
Thank you. I knew that it was going out. I had to review some data a couple of months ago, but I was so happy to see not only the paper come out after all these years, but also yesterday to see the hashtag. So I do share that feeling when something finally comes out.
Yeah.
So tell me a little bit more about your experience at Johns Hopkins. You mentioned you were a lab technician. What drew you to working in the lab, or what were you working on in that position?
Yeah, so at that role, I was working mostly on allelotyping, look at gene alterations in human prostate cancers. So, and I did a lot of microsatellite analysis mainly, looking at try to map down the gene responsible for prostate cancer. And I remember that because I wanted to be a medical student someday, I volunteered to do a lot of autopsy, to collect the samples, to be able to come back to the lab and analyze the samples and so on. So I still remember that I did exactly, I think 22 autopsies. And these were patients who donated their body to science, who, every time they died, they would call us right away. Sometimes it would go in midnight or 2 or 3 in the morning to be able to get the fresh tissue and do the whole, the whole autopsy, basically.
Wow, that is absolute dedication there. And so this
my boss was an MD, so I wanted to impress him to get a good recommendation.
I'm sure you did. I think at the same time you also realized that you do not want to be an MD.
Right. I did try to force myself to have that interest, but I couldn't influence myself.
And this was during undergrad at Johns Hopkins?
Right.
Or?
So by that time, I already finished my undergraduate.
Okay. And then what happened next after the undergraduate? You mentioned that very early on, you'd realized that you wanted to do a PhD, but how did you end up in the PhD program? How did you pick it?
Yeah. So basically, my life evolved around my families and kids also, in terms of my professional lives. So at Johns Hopkins as a technician, I got married, and then I had 2 kids, and then I realized that I just could not support the family with the salary of a technician in academia. So I decided to look for a job in industry, and that's when I found a job at DuPont Pharmaceuticals. Mm-hmm. Well, the pay was still not much, but it's still more than double what I was making at Johns Hopkins as a technician. So it was very helpful. And that was my start of the industry at DuPont and doing high throughput screening department. I think even then, I remember that my target I was working on was a CDK2E for cancer.
How cool is that? And you actually, thank you for sharing that experience, because this is something that typically people don't talk about, the fact that you actually do have a life outside of science and you have a family. And a lot of times, at least here in the US, people have PhD students and also postdoc have kids later and later than compared to Europe, for example.
Right.
And it might determine where do you go. Yeah. I don't know if you know Brian Airy, who's still at BMS, but Brian, during our podcast, he was mentioning the same thing. He actually ended up taking an industry job out of his postdoc because, 1, he was interested in industry, but 2, there was a family to be supported. And that's how we kind of ended up in drug discovery there.
Yes.
So it's a route. So you mentioned DuPont Pharmaceuticals and you mentioned GPCR. So when do GPCRs come into your scientific life?
Right. So when BMS bought DuPont, actually, I came to BMS 2 months or so before BMS decided to buy DuPont. So I already, at that point, moved over to DuPont, to BMS in Wallingford, Connecticut. That's in neuroscience. And they liked my high throughput screening experience. They wanted me to do, they brought me in to be able to do these classical radioligand bindings faster than traditional methods.
Mhm.
And so when I got in, I focused mainly on trying to optimize the process and try to get those, really basic primitive assays to try to get it more high throughput, basically.
Mhm.
And it's at that point that I started to learn more and more about the pharmacology, and I knew that there's a lot more for me to learn. And the more that I learned, the more I realized this is what I wanted to do. Because the receptor pharmacology is just so complex that it's not all about just testing compound and getting you an IC50. So
Yes.
So, my boss was from a good lab in Vanderbilt, and he was a GPCR expert. And I think his boss, who was the head of the department at the time, was also a well-known traditional old school GPCR expert. I think he was from UPenn. He was, I think, Perry Molinoff. I remember that name. I think he's probably long retired already. And so I was in a good GPCR lab, basically at BMS. And that's how, the more I learned from my boss, the more I realized that I had so much more to learn.
Absolutely. Absolutely. So you're joining BMS, and then DuPont gets purchased by BMS. So I'm assuming the colleagues from DuPont ended up in your circle back. And you mentioned GPCR pharmacology and doing binding assays. And this morning I was thinking about binding assays, and I have not done binding assays in about 10 to 15 years. And we used to do them the traditional way on the membrane. And I do remember working on CXCR7 and EMD, small molecule binding. And this morning I was thinking about okay, temperature, do you do it at 4 degrees? Do you do it at 37? And how do you actually track that interaction? And it's not a trivial assay to run, but still, isn't it beautiful when you actually see that saturation curve?
Yeah.
Or you see a displacement of your tracer? I think it's just wonderful.
Right. And one thing that led me to have a lot of interest in GPCR is the simple radioligand binding itself. And I remember my boss directed me, helped me design different binding assays to answer certain questions about allosterism. And I said, wow, that's a lot you could actually do in this simple assays, these basic assays, to really answer a lot of very complex questions. And so I think from then on, my career started to evolve. I said, I got to go back and do my PhD. And so, but then I realized I cannot, because I still, and now I'm a single income person, supporting now I have 3 kids.
Yes.
Yeah. I said, I got to find a way. I really want to get a PhD. I got to find a way. So then I basically tried to design my way. And basically I did my PhD in the non-traditional way, where I tried to get BMS. And then I also looked into Yale and UConn, the 2 institutions I looked at, I was looking to. And then Yale said, yeah, you can do the PhD here, but you basically have to quit your job because you will have to do exactly everything, the same thing as everybody else. You have to do rotations. You have to be here when everybody's here. You have to do presentation. And I was thinking, I cannot do that. I have to.
I have bills to pay.
Right. I had to still, have my full-time salary, and I had to find a way to do it. And so then I look at UConn. Then UConn actually gave me the flexibility to do my PhD, my own time, basically. And so I got the head of the department at BMS to agree with the UConn head of the department to allow me to do basically all my research at BMS, and all the research will be funded by BMS. And I guess even the tuitions, whatever the courses I had to take, was also paid for by BMS. So basically I got both institutions to agree the way I want to do PhDs, But then BMS said, well, you can, but we don't expect any less from you. You still have to produce the same quantity of work for us to maintain that full-time salary. And then you can't just require me to be there, I think once a month, actually once a week, to for the presentation, either as a spectator or to present to the group. And so that's basically it. And but then with me already having a lot of experience in the industries, a lot of lab experience, I basically designed my own PhD program to make it efficient for me, so that I would not waste any experiments like the traditional graduate student who actually would do a rotation and try this, try that, and they ended up doing so many experiments that not much of them actually was being used as as part of the dissertation or publication. So because I had so much experience, so much technical experience, I knew what experiments will work and what will not work, and what is practical, and so and what is not. So I didn't let my fantasy about the curiosity to basically do the driving, but I actually balance it with the practicality. And so by doing that, even though I'm I work full time and doing PhD full time basically, because there's no such thing as a part-time doctoral programs, but because I designed the program, my program so well that I actually was able to finish all the lab work and everything needed to complete my PhD in 3 years, rather than, and basically much faster than a traditional PhD student. Although, I did not defend my PhD until 5 years after that. Mainly because I think during that time 9/11 happened, and a lot of companies started to lay off people. And so the job market was very bad, and I did not want to defend my PhD and become overqualified for my current positions. So I try to sit back and relax and just slowly write my dissertations and just wait till the timing is right and then defend. So that's how I got my PhD.
That is phenomenal. I don't think anyone on the podcast has ever had a story like yours. So let's track back. I have a couple of questions. I think so. A lot of the audience of the podcast are either PhD students or postdocs, and I always like to ask questions from our guests about advice and to get a sense of how did they manage some of those accomplishments. And so just to make sure I get this, you were able to convince your boss at BMS and the director at UConn to let you do this. And BMS, and did you have a mentor or a PhD advisor at UConn, or was this done at BMS?
So they set it up so that I would have a co-advisor at BMS and another main advisor at UConn.
And the project that I'm assuming you dreamed up the project as well.
Yes. So at that time, I was working on CGRP receptors, antagonists for migraine. Actually, the compound I worked on and I optimized actually in the clinic right now, it was by Biohaven Pharmaceuticals that when BMS shut down, they in-licensed that compound and further developed that. And now it's Nurtec, and that's the reason why Pfizer bought then Biohaven to get that migraine medication. So I was working on CGRP receptors, and I was looking for antagonists. But then I decided I wanted to stay on CGRP receptors, and then I tried to design that so that I can still work on the same target, but looking at a different pathway rather than look at the signaling pathway. I look at the receptor desensitization. And look at and then compare with the different classes of that whole family of CGRP, amylin, adrenomedulin receptors, and look at just focus on desensitization rather than on activation. And one of the risks for that is that because I was working on the targets that for BMS, the risk is that I might not be able to publish that. But then I was still able to convince them. I said, I will make it so different enough that, even though it's the same target, it's a totally different story. The story is not going to be captured. It's not part of our drug discovery process, basically. And at that time, CGRP itself was already been presented a few times at SFN as a target that BMS was working on. So it would not be something news if somebody found out that we worked on CGRP.
That is phenomenal. I wish I were, I were able to hear that conversation, or how you got to convince everybody to say, well, actually, this is what I want to do. How about you? How about you do it?
Yeah, and there's a lot of there's a lot of leaders at BMS that I talked to. This is the head of the department and the next level down, this executive director that said, they said to me the same thing, basically, Sokhom, I don't know why you want to do this. You're gonna educate yourself out of a job. So your job is so secure now. and I said, well, it's okay. Let me deal with that. I'm willing to take the risk. And then they said, okay.
I love it. I love it. And plus, at the time, you were able to use. It's the same target that you were working on the drug discovery. So I'm assuming you had the assays, you had the setup, you had the tools, the HTS experience you had that allowed you to surgically plan that PhD work and just run with it.
Yes.
I love it. I love it. I don't think anyone I've ever met has done anything like that. Interestingly, I was having a conversation with someone who just finished their PhD and they wanted career advice. And they were so focused on saying, oh, I have to do a postdoc in academia. And I kept telling them, no, you have to decide what is your goal. What do you want to achieve? What's the lifestyle you want to have? And then find a path that leads you there.
Exactly.
And so, but it's such a difficult thing because, especially in academia, and I was part of that, let's say, cohort of people where I joined the lab for a summer internship. I did this in Montreal, so we didn't have rotations. You just pick a lab, you spend 3 months there, and I ended up picking a GPCR lab because I actually wanted to work with Michel Bouvier, who then referred me to a chemokine receptor PI who was just joining. So I ended up doing 9 years in the same lab. I did a summer internship, a master's, and the PhD. But at the same time, all I could hear at the time is you have to do, once you have your PhD, you must do a postdoc. And it's not true. It depends on what you want to do.
Right. So for me, I did not have to do a postdoc because I had a lot of pre-doc experience.
Yes, yes. And it's a very unique experience. I love it. So, you mentioned doing your PhD work in 3 years, taking enough time to graduate because of all the insecurities of the job market. And then you graduated at some point. And then what happened? What was the next step for you?
Yeah, so as soon as I graduated, I basically got a position for me at Novartis.
Okay.
Novartis Ophthalmology, basically they hired me also for GPCR targets in ophthalmology.
Mm-hmm.
So that's basically, Novartis is the only company that is not neuroscience, basically, but it's still GPCR focused for me.
Okay. Okay. And did they reach out, or did you look for a new position, or how did that happen? Because sometimes there's very interesting stories when a job transition happens.
Right. So here's the story. So I was looking after I got my PhDs, and the main reason is that when BMS allowed me to do PhDs, they said, Sokhom, you can do it, but we cannot guarantee you a job after you finish. We cannot guarantee you a PhD level position.
Okay.
But you are welcome to look after you finish.
Mm-hmm.
And we will not fire you based on the fact that you'll be overqualified. So they promised those things. I said, that's good enough for me. So I still remember I saw a position in Novartis and I applied. And it's just such a perfect match. And I applied and I said, I'm going to get a call for this position, for this role. After weeks, I did not get a call. And so then I said, actually, you know what? I know somebody there. And so this would be this story here is actually advice for young scientists. So I know somebody there, and this is actually a guy named Jihu Zhang, who worked with me at DuPont Pharmaceuticals. He's actually the guy who actually wrote the Z prime factors. He came up with the Z prime factor. That he wrote that factor, the paper there while I was working with him at DuPont. And so anyway, so I knew him. I sent him an email. I said, Jihu, can you do me a big favor and just forward my CV to the hiring manager? I remember doing that in the morning. And then basically like 3 hours later, I got a call from the hiring manager. And the reason for that is that I did not find that out until I actually joined Novartis. Anytime we had a position open, there were thousands of resumes in the inbox for that role. The hiring manager could not go through all of that, and so they just don't have time to go through all that. And if they need the help from the HR person to help filter out and sort out some resume, a lot of times they eliminate the wrong candidate. So then my take home message and my advice for people based on this is that it's important to network, important to know somebody. I mean, you don't need much help from your network. All you need is just a simple forward of your resume, and it makes a big difference.
It absolutely does. And I think a lot of times, especially people in academia, are afraid, or maybe more junior scientists are afraid of this word networking. It sounds like this big lifting, like you have to, do something super heroic, when in reality, it's not. We are networking right now.
Right, yeah. And networking, people think that you actually have to try to get to be buddies with somebody. Networking just means that you passively keeping in touch with people. For example, people you work with, 5-10 years ago, just every year, just send them a happy holidays email. Let's keep up your network going already. But don't reach out to people 20 years later when you actually need a job.
Exactly. That's not the way to go. No, absolutely.
Right.
I have a friend, and he is actually he coaches individuals but also companies, and he was talking about one of his former bosses when he was at a financial institution, and said that person actually knew everyone's name. They knew everyone's spouse's name, birthdays, and kids' names. So you would get a note. And so the story goes that my friend Rick gets a phone call from this person, and says to him, "Oh, hi, Rick, happy anniversary." And it turns out that Rick had forgotten about his anniversary. So this person actually reminded him and saved the day.
That's awesome.
So be that person who calls up and reminds and wishes you a happy anniversary. Lovely. So you're now at Novartis. You have a PhD level position, and what are your obviously working on GPCRs? But how did this role evolve from your role at BMS?
Right. So the biggest difference is that I noticed that at BMS it was basically a non-PhD level position, and I remember in those years, 8 years there, I was sitting at project team meetings. I had some ideas, trying to speak up at meetings, and people would speak over me, and people would ignore me because I didn't have a PhD. And whatever, they would, and then I would then I would get the message, and I said, okay, then I'll be quiet. But then the big difference is now that I was at a Novartis, a new environment, as a lab head, I'm sitting at meetings. And if I have an opinion, an idea about something during project team meetings, I would just clear my throat, and people just get quiet and just try to listen to me. I don't think I became a lot smarter, as soon as I got my PhD versus a few years before that. I'm still the same person. I gained a little more knowledge, but not that much more.
And I think maybe it comes with confidence as well. Where you get a little bit, the more experience you get, the more confidence you get. But I think the more you age as well, the more you feel like life is too short. I can't just not say what I think,
right.
Especially if it can help the program.
Yeah.
I love it. And so you're at Novartis, you have a PhD level job, you're taken into consideration when you clear your throat and people listen to you. But when was it, if you can maybe tell us a little bit more about what kind of GPCRs did you work on there? What was the context of the work that you were doing at Novartis?
Right. So mostly I worked on, I think, 3 or 4 different targets on GPCR, looking at for ocular diseases. And so mostly for AMD and glaucoma are the 2 main ones. And so and focus mostly on functional assays, but I also established some radioligand binding lab to able to spot check and verify and quality control the data. And so the main difference is just so packed, I mean, so little space we got, and I was very heavily focused on the lab and the bench, even though I was already a PhD level. A lot of work, a lot of hands-on work for me also.
Okay.
So it's very intense. So all those times at pre-BMS to BMS, and I had, spent so much time in the lab. It's actually the most fast-paced big pharma environment I've ever been. It was at Novartis. And so it was just to the point where I was just so focused on the technical aspect of things that I was just saying that I still need to get out of this technical focus positions. And then I was able to find a leadership role at Alkermes to actually be the head of in vitro pharmacology, where I would build a team from the ground up. And that actually shifted my scientific career from being a lab head to become a leader. It was at Alkermes.
I love it. And I know you and I offline in a different context talked about the team you built at Alkermes. But what I would like to hear about a little bit more, and this is mainly for the audience, is how did you approach your leadership role? Because obviously you've seen the good and the bad. I mean, we've all had the good bosses and the bad bosses. You know what not to do. But how did you navigate that new role of leadership?
Yeah, so actually, that's a good question there. So I came from, by that time, I already been exposed to a few bad bosses. So then when I got these leadership roles, as soon as I got the offers, I just could not sleep. My energy level was just so high, and that I would just lay down in bed, before my start date, thinking about my strategy, thinking about how I would hire people, the type of people I would hire, how I would treat people, how I would establish my managerial styles to prevent a lot of negative impact that I've had when I had some toxic bosses along the way. And so I started to read some advices, online here and there, and from a book, just to try to develop my own way to lead a group. And when I got a chance to do that at Alkermes, even I would just say, even like the first half a year, I did not sleep much, and my energy level was. I mean, even though with a lack of sleep, I could not feel that I was actually that it affected my health in any way, because I feel so much energy all the time. And that came from my excitement of that new role, to be able to have that so much empowerment to design my own team, to hire my own team, to basically do anything I wanted to do. They trusted me. Alkermes at the time was Alkermes, I think it's an older company that started since 1981, I think. But the company had, they had R&D, but mostly D. They were known for their development in terms of formulations and basically, so research was not much. And so basically I was hired to put the R in R&D. So I really expand the research component.
I love it. I love it. And then I'm assuming also still working on GPCRs at
Yes.
Alkermes as well. Yeah, it's interesting because people don't typically. So I get a lot of emails from PhD students and postdocs telling me, well, I want to go into industry, but I don't know which companies work on GPCRs and what does that look like. And I'm curious to hear about the landscape of GPCR research in industry. Since you've been in industry for such a long time, have you seen it go up and down from a GPCR, from an interest towards GPCRs?
Right. When I started, yes, exactly. When I started BMS, I think GPCR was the hottest field at that time. Actually, when I started DuPont, HTS was the hottest. And then when I started BMS, it was during the time when GPCR was hot. And then it actually, I think 5 or 6 years after that, so a lot of companies started shutdowns, the neuroscience, and shut down GPCR, ion channels, and now it's starting to come back. So it's a wave, it's a cycle.
Yeah. And being on the Dr. GPCR podcast, for us, GPCRs always are of interest. Definitely. Definitely. So now you're at Alkermes, you get a chance to build your own team, you're developing your own leadership style, your own managerial style, and then what next?
Yeah, so I focus basically on combining the classical radioligand binding, classical receptor pharmacology with modern pharmacology. So I started to looking, and even in terms of my strategy of profiling compound, profiling molecule. I think at Alkermes we're working on. I've worked mostly on opioid receptor system, got pain, and so we try to not only focus on the mu opioid receptor, but also look at the biosignaling component of that. So I work with a team of business development to in-license the biosensor technology from Domain Therapeutics at the time. So that was the technology. Then we brought that whole technology in-house, and we actually generated so much biosignaling data using that technology, combining with the radioligand binding lab that I established in-house from the ground up there. And so the combination of both actually was very powerful.
I love it. I am Brett biased because I actually, at the time when I started my master's, Michel Bouvier was developing his Brett biosensors. And it was a time where you can just go up the hill, because we were down the hill, and you get a sample of DNA and you walk back to your lab and you just use the DNA. So very much Brett biased on that front. And you can generate so much data with these biosensors. And I find it fascinating.
Yes.
Wonderful. So after Alkermes, you got the opportunity to start and build Cerevel.
Yes. So I still remember in the back of my mind, I said, wow, this position at Alkermes is just so exciting. I just never thought that, I would ever say in the future at any point in my life that I fell in love with the job. That was actually the first time I fell in love with my role with the job at Alkermes. And I remember thinking, I said, wow, just building the team from the ground up with so much empowerment made me so happy and so passionate about science. I wonder what it's even like to build a company from the ground up. And I had that in my mind. And then I remember the reading about Pfizer shutting down their neuroscience department. And they intended to actually reestablish a new biotech company from that portfolio. And I said, "Oh man, this is awesome! I should keep my eyes open for that, for that opportunities." But then, years passed by, and I totally forgot all about that. And then I got a call from a recruiter for the opportunity at Cerevel. I said, "This is the thing that I was waiting for," and I totally forgot all about it. I feel like this is what it's just meant to be.
I love it.
So,
love it.
Yeah, so I got recruited to join Cerevel, and that time, I think when I was interviewed for the position, Cerevel has less than 20 people.
Wow, and so this was an opportunity for you to start all over again, with a blank page, to build out a new team, an R&D, GPCR from in vitro pharmacology and biology team. What are some of the take-home messages, some of the things that you learned at Alkermes that you were able to take with you to Cerevel?
So yeah, what I learned from Alkermes is that for a team to be a successful team, to be in terms of quality of data and the amount of data we would produce, and basically a nice, healthy team in general, you need to build the right coaches. And so with my team that I built, I focused on the coaches, and I remember that my team was known within the company as the happiest team at Alkermes, and that's the feedback I got from multiple people. And to me, that was very important to me, because I want people to come in to work every day passionate about what they're doing. I don't want them to come to work because they feel like they're under pressure to get things done, based on a certain timeline, strict deadline. And when people are passionate, they're just automatic, and you don't have to push them. Right? They just come in and just focus on getting the work done because they're passionate about their work. And so when I started to do the same thing at Cerevel, my main focus was to build the culture right. But then, because the company is just a startup company, I also wanted to play a bigger role to not just build the culture of my team, but also to help build the culture of the companies. So even though we start to hire more people with different functions like DMPK, clinical, and talks, all these other areas of chemistries, I wanted to be involved in the hiring process. I wanted to be involved in who we bring in to be part of the Cerevel team, because I wanted to make sure we can avoid, bringing a toxic person. My philosophy, it doesn't matter how genius a person is. If the person is toxic, I just don't want them to be part of a team.
No, I agree, and I think it only takes one bad apple.
Right.
Especially when you mentioned joining Cerevel and they were less than 20 people. It's a stage at a company where everybody knows everyone. That's the opportunity for actually figuring out who has how many kids, what's their name, their birthdays, and when is their anniversary, in order to then be able to network with their colleagues. So I think it's very important.
Yes.
All right. So I have to ask you this. What is your favorite GPCR?
Well, actually, I don't really have one, but I just love GPCR in general.
Mm-hmm.
And the reason for that, I remember talking to Arthur Christopoulos in one of the conferences, and he said that he just loved studying the behavior of different molecules in different GPCRs, and that was also my interest. But then he was saying that the problem with him was that he don't have enough access to all the different molecules the industry has. And then I was thinking to myself, well, that's my advantage. I have all the access to different molecules everywhere I go. So I just my passion is actually be able to see different molecules and to see how the different molecule can behave so differently, even through the same receptors. And so that was my passion. I just can't wait to see more, molecules who have a distinct profile hitting the same targets.
Yeah. I think it's fascinating. And what I find fascinating with GPCRs and GPCR signaling is not only the molecule on the receptor, but just the lipid membrane, the lipids in the membrane, and how that membrane composition even changes the receptor function. Yesterday, we were talking with Tom Sakmar and Ilana about RAMP proteins. And I was like, wow, I mean, it's a single transmembrane protein. There's 3 of them. And people are like, yeah, well, RAMPs. Well, no, actually, they're really important and interesting.
Yes. The whole class of receptors is the most complex and the most interesting. Where RAM is not just known as, it's not just an accessory protein, right?
Exactly.
I still remember when I found out that because just going back to BMS, when I was working on CGRP receptors, I remember we started the program from the beginning before we had 2 compounds. Right? So the in vivo model that people developed was using the endogenous ligand, using CGRP to develop in vivo animal models. And then we started to do in vitro profiling. We found a lot of potent molecules, but then none of these molecules actually had any efficacy in vivo models. And we didn't know that, and no one had any clue until we saw the paper published from Merck that it was just due to RAM1. And not only is it just due to RAM1, it's due to a single amino acid on RAM1.
Yeah, this is what I absolutely find fascinating about GPCRs in general. I mean, just one amino acid changes everything.
Yes.
That's fascinating. I mean, even the science paper we were discussing, we actually studied mutations in GLFI2, and mutating one amino acid in GLFI2, and it can be a different amino acid depending on patient populations, actually impairs the interaction of the G protein with the chemokine receptor. And you get all sorts of immune deficiencies and all sorts of infections.
Yeah, and that's and it's because of that example. Every time I would have a discussion, 20 years or so later, when people always said, "Oh, we expect the ligand to have similar affinity in animal because the homology is 90% or 89%. I said, no, it's just one amino acid, all it takes. I don't really care about the percent homology, just get the data.
Exactly. Just put it in the animal.
Yeah.
Let's see what we get. I love it. Top 3 aha moments that inspired you and that shaped your career trajectory.
Yes, I think the aha moment for me is actually one keyword is empowerment. I did not change the way I work, the way I lead, or I could not see my full potential until I was at the environment at Alkermes where they empowered me to do everything. They trusted me, and I designed the whole thing. I had a boss who was hands off, and he trusted me to just, let me do things my way. And that empowerment was the key thing that actually changed my whole career. And when I lead people, I also wanted to make sure that they have that opportunity to be able to make their own decision, make mistake, and learn from it, and to be able to have that empowerment to think freely rather than being micromanaged and being suppressed by a boss.
Yeah. And I think that's very important as well. And one thing we have to say this because this podcast episode is going to come out definitely after next week, is that as of next week, you're going to be joining Superluminal as head of biology, and I'm very excited to be working with you.
Me too.
All right. Any advice? Actually, are there any other aha moments? So empowerment that allowed you to really spread your wings, let's just put an image to it, and become the leader that you've always wanted to have. Any other scientific aha moment?
Yes, I would just say, follow your passion. And, my passion was GPCR. I stuck with it, even when GPCR became unpopular for many years, I just stuck with it. And to me, it's the good. I've been telling some of the people in my lab now, I said the good thing about the fact that GPCR became unpopular for the past 10 years or so is that if they stopped producing new scientists to focus on GPCR expert. For example, immuno-oncology became more popular and people shifted that way, right? And so for me, I had, and during that time, the older GPCR experts, they retired and they did not develop new ones. So I have a lot less competition now. So it's great for me.
Less competition, less molecules to compete with, more targets to work on.
Yes.
I love it. We talked about a little bit about advice for junior scientists, and one thing you mentioned is really networking and just reaching out to people, being human. Any other advice to scientists who either want to contribute to the GPCR field in general, but maybe want to contribute to the GPCR field in industry?
Right. I think if anyone who actually wanted to be an expert in GPCR in general, it'll be in the industry, because then you're not focusing on one specific receptor. You actually will expose yourself to so many different molecules, different ligand, different receptors, and just so, because we're shifting from target to target, even if you're working in one company. So you have a broader exposure and to really learn the whole field. And if you can see that receptor pharmacology textbooks getting thicker and thicker, right? There's a lot more and more to learn.
Absolutely. And I think one advantage of being in the field in industry is having the opportunity to use the latest technologies, the opportunity to call up people who are KOLs, who are experts, and say, "Hi, I have this problem. Can we have a chat about it?" And I'm going to mention Terry, because Terry not only is wonderful, but he says hi. By the way, I spoke to him on Monday.
Awesome, thank you. Yes, when I met Terry for the first time, I think about more than 10 years ago, I told him, I said, "Terry, you don't even know it, but you were my teacher. I developed my expertise by reading a lot of your paper."
Yes, absolutely. I think he's not only wonderful as a person, but he's the smartest GPCR pharmacologist I know.
Yes.
He's definitely out there. Any other advice for scientists? Anything? I think one thing that's missing in academia, and I know we're talking about your experience in industry, but one thing that's missing in the training in academia is the leadership skill. So the skills that you learned and that you applied by working with not what by learning what not to do could be skills that PIs, brand new PIs could be using, because you can have a bad boss anywhere in any
Right.
field, no matter what
Yes.
you're doing. So
Right.
any advice on what or how to go about it, where to start?
Yes, I think so in academia, especially in the PhD program or postdoc, scientists were not trained to network. They just trained to focus on a small section of their bench, trying to get something published, right?
Yeah.
And so, but you have to actually train yourself to go out of that comfort zone or uncomfortable zone. I mean, you actually, for example, had a chance to attend scientific conferences. Don't just go there and present your poster and just hang around with the people you actually know from your own lab. Just go around, chat to people, talk to people, get their contact information, reach out to people, and just chat science. Just go out to strangers. Don't just hang around with the people you know. That's a
Avoid the
key
people
thing.
you know.
Yeah, avoid the people you know. Go to a place where you actually had no choice but to actually have a small talk with somebody.
Yeah, and that's a difficult thing to do. Whether you're in, I think most scientists are introverts, which makes it even more difficult. But even if you're an extrovert, it's very difficult because you like your comfort zone. You're like your friends. You sit with everybody you know, but that's not where the magic happens. It's
Right.
when you talk to other people. You never know who you're gonna meet.
Yes.
All right.
And the best networking advice I had was I forgot to mention was this actually how it changed my career also. It's transitioned from Novartis to Alkermes. And I was in Novartis, just stuck in one position doing the same thing, for so long. And Novartis had a mentorship program that was designed only for women, and because they want to promote and enhance women's leadership and women's and so then that same years, I think that was my last years at Novartis, that they started to open up for men also. And when they did that, I signed up. And then when I signed up, I chose, rather than choosing another scientist as my mentor, I chose the head of diversity and inclusion to be my mentor. And my first meeting, I feel like he could see through me that I was frustrated about something. And so he said, Sokhom, I'm going to have an assignment for you. You're going to focus on networking. And I'm going to give you 5 names here to reach out to and just have a coffee chat or lunch, whatever. Most people will eat lunch even if they don't have time to talk to you. At least, use that time to, to meet with them and chat with them. People at higher level, different level. And then each time you meet one of those, ask them for a name or for at least 2 or 3 names that they can recommend that you could reach out to for networking, and just keep on doing that. Each time you meet somebody, ask them for somebody else to meet. And by the time I did that, I changed myself from being an extreme introvert to an extrovert, basically.
I love it. I think this is fantastic advice, because you it's like eating an elephant bite by bite, because you only pick 3 people, but then each of them, it exponentially grows. But by the time you get to the 3rd or 4th level of people you're meeting, you've done so much talking and so much networking, and you get to know so many people.
Yes.
It's very slowly getting into that comfort zone. I love it.
Right.
All right. So the biggest, most difficult question of the podcast is, what should we title this episode?
I think just based on how I evolved my career, right, to be where I am now, I did it non-traditionally. I knew what I wanted to do. I knew I wanted the highest level of degree, and I knew I wanted to be a scientist. But I could not even, in my early days, I could not even imagine me being a leader, because I was so shy, being an introvert. But anything's possible. I was just saying, just stick with your passions and keep on improving and evolving your career. Don't let your weakness define you. You have to actually try to overcome it rather than accepting it. And networking, being an introvert, was something that would get me sweating thinking about it. And even giving a presentation, it would get me sweat thinking about it. Yeah. But then it came to a point where, after so many years, giving a presentation is like giving me excitement now these days, because it's an opportunity for me to show off.
Yeah. Absolutely. Absolutely. Yeah. No, I love it. So I actually captured, don't let your weakness define your life or define your trajectory or define who you are. I think it's there, when you have problems or issues or anything, they keep on coming back because you need to overcome them.
Right.
But it's painful.
Yes.
It's absolutely painful. So what do you think of this title? I think we should keep it.
Yeah, that's good. It's good. Yeah, good title.
I love it. I really appreciate your time, Sokhom. Don't go anywhere. We're going to stop recording. I'm going to get your first impressions after we stop recording. And as I always tell people, while we're recording, we're going to let everybody wonder what we talk about after we stop recording. Wonderful. Thank you again for coming.
Thank you for having me.
Huge thanks to Dr. Sokhom Pin for joining us. His story is a powerful reminder that your path doesn't have to follow anyone else's blueprint. Whether you're launching your own PhD while working full-time or transforming a team through trust and empowerment, Sokhom proves that you can evolve without losing your passion for science. To stay connected and amplify more GPCR voices, subscribe to the Dr. GPCR podcast and consider leaving us a 5-star review on Apple Podcast. Visit terrykanakin.com to explore Terry's Corner and level up your own pharmacology skills. And check out the episode description for more links, including links to Celtaris Research, our brand new partner. Learn more about their fluorescent ligand tools and how to apply them for your own research. And as always, thank you to the Dr. GPCR team for making this episode possible. Because receptors don't work alone, and neither should you. Until next time, stay safe.